Call it. Smile at the skeleton.
Boy, it's high-level instructions today. Let's turn to the line side, and up there at the top above that red line, please put in the upper left corner, last name, print your last name, comma, first name. Okay. So the instructions get more complicated. Flip the card over. In the upper left-hand corner, do the very same thing. Last name, and then first name. You'll see we use these cards over throughout the semester to try to decrease instruction increase. Any questions before we get started? Anything you'd like to go over? Okay. This won't take.
And then
Okay, so this the blank side, this is how I want you to set it up right now. Last name, first name, 1, 2, 3. You can make it pretty small because this is going to be multiple choice. You just put a letter in. And then in the upper right-hand corner, you can see a sideways 4. We're going to use the rest of this card for something else. Okay. Hopefully, like I mentioned last time, we're going to look at this boy right here. And you can see that this diagram provides choices for the items to come. So many medications are utilized to block or inhibit elements of the eicosanoid synthesis pathway. And that's what this is. Where do the following exert their actions? We could try inhibitors for number 1, please. So the answer will be A, B, or C. Okay, next one. Non-steroidal anti-inflammatory drugs, NSAIDs. 2 examples of many, aspirin and ibuprofen. A, B, or C. Okay, and then, not a surprise, number 3. Steroidal glucocorticoid drugs such as prednisone. Where does that exert its actions? On this pathway. Now the one that's going to take a little bit longer, I'm going to give you about 5 full minutes to do, is number 4. This will take up most of your. Please illustrate and label the bidirectional homeostatic sensor effector negative feedback loops for glycemic control for controlling plasma glucose. So tell me, what is the cell type? What cell is it for the sensor? Tell me what hormones are involved in wherever you put them in your loops. And give me one effector cell types. There's many. So basically, illustrate and label. Yeah, you don't have a whole lot of room, but that's kind of the idea, is do things a bit succinctly and drawing out the homeostatic control groups for glucose. As you're going to be thinking about these the rest of your life. Okay, we won't always get this option, but would you like to confer with your table colleagues about any of this? Oh, okay, okay. So, this table you're together, that table you're together. Stay within your tables, closed book. You, two in the back, guess what? These are friendly folks up here. Come say hi to them. Now, you know, a big part of professional interactions is listening and giving advice and trying to differentiate what's good advice. I don't know. Talk amongst yourselves. Do this for at least 4 minutes.
This is just the bi-directional through negative feedback. So like if glucose is too high, the glucose is too low. What is that? Beta cells or something? Right. And then the sensor is negative feedback.
It's like you're sensitive to it is.
Yes. So glucagon is the effector. That? Well, this is old. So then glucagon is the effector, and then that releases the glucose. And then that is a negative feedback. The low amount of blood sugar. There's receptors that see that there's less glucose. Well, I guess the absence of binding to those receptors says, hey, there's less. Yeah, and then that's negative feedback to glucagon. What do you mean? The lack of glucose acts on the liver to say that, oh, we need more glucose in the blood, so it releases glucagon, which helps make more glucose. I was like looking at that.
Take 1 more minute to finish up. 1 more minute to finish up.
We both did it a little bit different. And because I think we're the same thing, it's just a little bit rotated. Yeah. I think we'll accept it.
Yes. Insulin. Uptake of glucose into cells to reduce blood sugar. That's a negative feedback on insulin. So insulin is saying there's too much glucose, so let's take away. And there's less glucose that inhibits insulin. And that insulin is stopped being produced, so the glucose level is
I explained what I actually was doing because I don't know if I diagnosed the action.
Okay, let's hand them to the inside of the little tree. And you don't need to write who's all in the group today, that's fine.
No, it's not. It's a mess. It's on there. Just so. But you look at the text. So the no says do but steroids
Everybody started? Okay, now, we don't go, obviously, we don't go all quizzes like this, but there's a reason why you do this. And all of us have been in the position where you're studying or you're taking an exam or you ask a question and you realize, well, you know what you know and you know what you don't know. And yes, we're constantly trying to find out those things that we don't know that we don't know. It takes some outside entity to identify that. But you know when you go, I just don't know that, and it just eats at you. Cognitive psychologists have a name for that. It's called cognitive dissonance. And the body wants to relieve that uneasy feeling right away. And some of you probably, went, oh boy, I'm not sure about this. You had some cognitive dissonance. When you relieve that right away through interactions with a colleague, especially with peer-to-peer teaching and learning, that tends to go into longer-term memory much, much more quickly. And so, there's some of you who went, oh, I didn't really know this, and your colleagues helped you. Guess what? You helped your colleague by your colleague explaining it to you. That solidified the concept for them. You feel a lot better because you go, oh, they had an idea, but I didn't really know. But the bottom line is, you tend to take this away. You tend to remember this stuff. If we all had time, the way to give an hour exam is to give an hour exam and take everybody's answer sheet and then let people give the exam back and let people discuss among their colleagues and then write out a different answer sheet. And let's say 80% of your score comes from your first pass, 20% of your score comes from your second pass. The bottom line is you read how many times you take an exam and a week later you don't remember anything.
every time.
Yeah. Well, that's not going to happen in this course because the Unit 1 material is also in the Unit 2 and Unit 3. So I know this course won't be the same. But when you do those types of things, it stays with you. It transfers to long-term memory a little bit quicker. So we won't always do this, but this particular pathway, this suppository pathway, it is so incredibly important in clinical medicine. Yet in this course and many other in pathology courses, you're going to talk about this all the time. In this course, I'll only talk about it 2 or 3 times, and this is so important in clinical medicine. This little bitty thing, and what does this pathway do? What does this pathway do? What does this pathway do? How can we manipulate the output of those pathways with different drugs? And it is so very, very important. And this is something you're going to be living with, thinking about glycemic control the rest of your professional life. There is I think there's 35 questions, and I'm still trying. I have an incompatibility, but it will be taken care of today. This we put on Moodle, and for a backup, I'm hoping to get everybody's email address so I can also email this to everybody, the PDF, because tomorrow morning at 7:30 we'll do the old exam dissection for those of you who want to do this, and we probably won't have time to go through all 35 questions. Probably won't, but I'll try to make sure the questions we'll go over every single concept that will be on next Tuesday's exam. And I'll hope that you'll still go through the other practice questions. But with a 35, boy, I imagine that'd probably really take us a little more than 2 hours to actually get through all 35. But, and we don't have that much time. But what you're going to find is after we get through about the first third of this thing, we're going to have already covered a lot of the concepts because, and I'm going to be showing you, there's some things that I do like taking computer exams, but there are a couple things that you can do with paper exams that not all the computer exam programs are much better than they were just 5 years ago. I'm going to be talking about isolating automatic qualifiers tomorrow, like steroid hormones, signal to cells, what to do and when to do. But boy, there's an automatic qualifier right there, steroid. The whole question, if that says peptide, it's a completely different thing. That is, so I'm going to be really talking to you about slowing down and underlining automatic qualifiers. Not even looking at these choices, but thinking, okay, steroid hormones over here very, very quickly, and it usually only takes 10, 15 seconds. Jot down what you know about steroid hormones. Okay, they're lipophilic and hydrophobic. That means they don't want to hang out with water. That means they have plasma transport proteins, and they can go right through the membrane, and they will bind receptors inside the cell. Okay, and then you go through this. And so we'll go through this tomorrow. And no matter how many questions we get through, I hope that we'll be able to cover all the concepts, at least once, that you'll see on Tuesday. I can't guarantee it, but I hope we will. But I'm efforting to make sure that this will be on your information system, and hopefully I can email it to you so you can print out and have a copy to work with tomorrow. Also, what's going to be, and unfortunately Manuela's not well today, so she was going to help me, is there's an independent study, PowerPoint, going through G protein signal transduction. I think the students that have gone through that before for years now just feel so much more comfortable once they go through it. It's really not new information. It's just slide by slide, allowing you to see it stream, and you can stop it. So it's not too fast, it's not too slow. It's just right. It's homeostatic, right? A little bit later on, sometime this weekend, I'll post a learning objectives list, and okay, for this area, this is what I really want you to, before that Tuesday exam, be able to understand and do. Seems to me that there's one other, there's a simple sheet differentiating exocrine from endocrine, telocrine, paracrine, and autocrine. Really simple, and it's one of those things that you see once and you take it forever. And most of you probably are comfortable with those things, but in one little sheet and some simple diagrams, it's like, okay, no problem. I kind of knew that. And how, I'm sure you're like me. You know something, but once you see it, or somebody tells you, yeah, you're right, then you know it, and then you will believe yourself. And so much of professional training is believing in yourself, especially when it gets to national board exams. It's believing in yourself. Okay. We will have our old exam. We'll have our practice question dissection tomorrow at 7:30. And tomorrow morning, let's solidify. I want to be around for a couple of hours, at least late Monday afternoon. It turns out I am tied up until close to 5, so I won't be able to get here until about 5 o'clock. I understand this building's locked up. But I've also been told that there's some other options, and the roast lab might be an option where if I can get in there, Park myself, and I understand there's more at the board, so if you have questions, we can go through things. And I can certainly be here a couple hours on late Sunday afternoon, I believe it is. So I want you to at least have 2 shots of coming and asking me questions. And as always, when you come asking questions, bring a friend. That is so important. That is so important because that colleague is going to ask you a question that you didn't even think of and help relieve some of that not knowing what you didn't know. Okay, so we're going to finish up with action potentials today. And I did want to just mention to you that I was really hoping that action potentials could be on this exam, but it won't be because I told a number of people that the cutoff point for the material was going to be someplace from Tuesday. So where the cutoff point for the material will be, it's on page 151. There's this figure 617, but really it's the information on graded potentials. The graded potentials is the last information, and that illustration 617 is also very important. Now, there will be things on action potentials on this exam, but it's things that we've already covered. Today we're going to talk about the mechanism of action potentials, but I already showed you this slide, and I mentioned to you this is such an important slide to understand intimately. And this generic motor neuron or central nervous system neuron, where do you have action potentials? Where do you have graded potentials? You have action potentials possible because you have these types of translocation channel proteins. The graded potentials up there, they are made possible by ligand-gated channel proteins. A lot of information on here. You might not think and you might not appreciate it, but there's so much information that for this exam and the exams to come and things later on, if you just truly understand what's all on this slide, it helps you greatly in so many things coming up. Are there exceptions to it? Yep. And you'll learn those exceptions. So we talked last time about graded potentials. Graded potentials, once again, on this neuron, they're occurring up on the dendrites and cell body. Dendrites and cell body. We're going to turn our attention to action potentials, which really are formed initially in this area called the axon hillock. Action potentials will run down the axons, go across the synaptic terminals, and they will release neurotransmitter. So always contrast and compare. Graded potentials are generated up here. They can't travel very far, but they're constantly being reinforced by summation, and those graded potentials are all integrated here at the axon hillock. The axon hillock is an integration point. It takes all of that information. I like to think in this type of neuron, the dendrites and cell body, this is the information reception region. All that information is being integrated right here. And if it drives the axon hillock to a level called threshold, then you will generate action potentials that will propagate down this axon. But if the graded potentials are not great enough to drive that axon hillock to threshold, then this neuron remains quiescent and it's not going to release neurotransmitter. Okay. So let's very quickly, what we talked about last time is some of the rules, and I just remember the terms depolarization when that binder was up, repolarization going back down to resting membrane potential. If it goes beyond resting membrane potential, that's hyperpolarization. But graded potentials, those things that occur in several places, but what I just showed you with the neuron, dendrites and cell bodies, they're called graded because different amounts of stimulation will cause differing amounts of depolarization. More stimulation, more change in electrical activity. And there can be excitatory graded potentials. These are initially depolarizing. There can be inhibitory stimuli. And what do? They are initially hyperpolarizing. Hyperpolarizing. And the whole thing about these graded potentials is graded potentials send an electrical current down to, and the illustration I showed you before, the axon hillock, that's in a different place with other types of neurons. And all that information would be summed. If it's depolarizing, it would tend to turn on the rest of the neuron. If it's hyperpolarizing stimulation, it tend to keep that neuron turned off. We talked about summation. So a neuron, a patch of membrane that can form graded potentials, if you stimulate it a little bit, you get a little bit of depolarization, and there's many automatic processes that bring the membrane back to the resting membrane potential. And we're not going to talk about all those automatic processes. Does the sodium, potassium, ATPase exchange play a role? Indirectly, yes it does, but there's many other processes. And that is a huge subject area. And until you need to know it, we're not going to go through it. But if you happen to stimulate that membrane 1, 2, 3 times before those automatic processes can repolarize the membrane, you can see you have this increase in the degree of depolarization, an increase in the amplitude. Amplitude is the height. The amplitude is the degree of movement away from its resting point. Since this stimulation is occurring 1, 2, 3 over time, so this is stimulation from one source occurring over time, and it has to occur quite quickly. That's called temporal summation. You can also have this thing called spatial. Spatial means space. And here you'd have 3 stimulating events, probably from 3 separate sources occurring at the same time. So generally speaking, temporal summation, it's a volley of stimulatory input coming from one source. Whereas spatial summation, it's stimulation coming from 2 or more sources at the same time. Bottom line is you get the same type of effect. And both of these occur all the time. Yeah.
So for spatial summation, is it possible to have one excitatory signal and one inhibitory signal at the same time?
Bingo. And that's what we're really, when we take our exam, I guess I should mention this to you. When we take our exam on Tuesday, it's an hour exam. And I think it's 30 questions or so, plus or minus a couple. And so we'll take an hour to take the exam, and then we'll spend the rest of the time in class time. And that's what we're going to talk about, neuromodulation. Get this, let's say this is a neuron in your central nervous system. My body is the dendrites and cell body, okay? So there's excitatory neurons plugging in here, there's inhibitory. You have between 100,000 and 200,000 synaptic inputs coming into this one area. Some are excitatory, some are inhibitory. So yes, you can have both excitatory and inhibitory arriving at a patch of membrane at the same voltage changes. and ligand gated. Some pop open in the presence of a ligand, some close in the presence of a ligand. There's a lot of specific different ones in the body. But ligands control if this is open or closed. Voltage controls if these are open or closed. And I don't want you to think that there's just one voltage gated for, in this case, sodium channel. It turns out there's 40 or 50 different ones in the body. And there's something like 60 of those potassium ligand gated. Well, I'll get to that later on. So there's a whole number of different voltage gated proteins. So voltage ligand. So this is the way a ligand would work. Here we have the ligand binding. It opens up the channel. That causes, in this case, this happens to be one that allows sodium to come through, and that would cause depolarization. So action potentials are all characterized, well, most all characterized by a rapid depolarization. So this would be a type of action potential you would see in skeletal muscle and in neurons. And we see a rapid depolarization. And they don't last forever. Once the job is done, the membrane then repolarizes. Once again, these are communication signals. For muscles, neurons, and some glands, these little electrical blips tell that cell what to do and when to do it. And you don't want the signal to stick around forever, so boom. Once you depolarize, you've told that cell what to do, and then it repolarizes to get back to its resting membrane potential. So right now it's not such a bad thing for these types of action potentials. What's that depolarization phase due to? Sodium influx. What is the repolarization phase due to? Potassium efflux. There's this change, rapid change in permeability when the membrane is rapidly permeable, rapidly but very briefly permeable to sodium, and that allows the membrane to polarize. And those same voltage-gated sodium channels, they sense we've done our job, the membrane's been depolarized and those channels close. At the same time, there are potassium channels that sense what's going on here. The membrane's depolarized. Get it repolarized back to normal. So they spring open, and the potassium efflux repolarizes it back to resting membrane potential. So action potentials in skeletal muscle and cardiac muscle and neurons, very rapid, robust, upright depolarization phases. But repolarization looks quite different in different cells. Yes?
So if potassium repolarizes, if the membrane potential of that cell is not minus 94, is it then going to cause hyperpolarization?
It can drive it past it. Let's go through the next set of slides and see if it fits where you're going. Please ask again if it doesn't answer it. So depolarization, they look very similar. Repolarization, really very dependent on cell type, because you can have other things going on. Over here we have a calcium channel effect in a cardiac ventricular muscle. So in neurons, man, the entire action potential is just 1 to 2 milliseconds. Skeletal muscle, not much longer, 5 to 10 milliseconds. That's fast. That's fast. So action potentials, they all kind of look alike in these types of tissues. The repolarization phase, excuse me, I should say the depolarization phase, kind of looks alike. But the repolarization, it depends on which cell. Depends on which cell. Here's some concepts that I know you've heard before. Action potentials have threshold voltages. There is a specific voltage that will cause that sodium channel to spring open to allow sodium influx to allow the membrane to polarize. So let's say we're sitting in an axon hillock, and braided potentials are coming down from the dendrites and cell body. And here's a little bit of depolarization, but nothing happens. A little bit more depolarization at the axon hillock membrane, nothing really happens. Little bit more depolarization. That's what really happens. Down here is the strength of the stimulus. So, you know, up here in the dendrites and cell body, we have an increasing amount of stimulus strength, and the current that's finally reaching the axon hill, if you polarize it a little bit more, but when there's what's called a threshold stimulus upstream, it can drive that membrane to threshold, and then boom. It's like roll program. You have the automatic process of the action potential taking over. So you open up the first voltage gated sodium channels, and you get that rapid depolarization. At some point, both the sodium channel realizes we've done our job, and they begin to close, and the potassium channel recognizes we're depolarized, and they spring open, and then you get repolarization. So that's threshold. The other thing is these action potentials are all or none. All or none. And that even if we give a suprathreshold stimulation, the amplitude of the action potential is still the same height. The membrane is such that when you hit threshold, it's going to give you 100% and giving you a full depolarization phase. It's going to give you everything it's got to depolarize. So you don't see like a graded potential, the amplitude of the action potentials getting higher. So this is definitely one difference between action potentials and graded potentials. Now, this is kind of a scary slide until you deconstruct it. But action potentials also have what are called refractory periods. Refractory periods are simply periods of time following an action potential when the membrane is resistant to a second follow-up action potential. So, a refractory period. A refractory period is simply a period of time following an action potential when the membrane is resistant to forming a second follow-up action potential. There's absolute refractory period and relative refractory period. During the absolute refractory period, during that period of time, no way, no how can you generate in that time period a second follow-up action potential. Can't do it. Membrane won't allow it. But if you wait just a little bit longer after the first action potential, yes, you can generate a second follow-up action potential, but it takes more stimulus to do it. The membrane is still relatively resistant. Why? You actually have an increase in threshold, and that's what this is showing. So let's say we stimulate the membrane right here, and we're going to deconstruct this one at a time. We stimulate the membrane to threshold right here. If we would stimulate the membrane during the time period of the absolute refractory period, even with a suprathreshold stimulus, we do not see a second follow-up action potential. And we sure as heck don't see a second action potential on top of the first. Action potentials do not sum. Why can action potentials not sum? Because there's this absolute refractory period. You've never ever, in all your exposure to action potentials, seen an action potential on top of an action potential, like you saw with temporal summation. Didn't see it. Why? After you generate an action potential, there's this quiescent period in which the membrane is 100% absolutely resistant to forming a second action potential. So, absolute refractory period, you might want to, and I don't know if I put this in your notes, absolute refractory period ends, and the relative refractory period, same point right here, with the opening of the H gate. With the opening of the H gate. Did I give you that? Okay, good. Right now that might not mean anything to you, but will here in just a couple minutes. So let's deconstruct this and look at it. You'll need to tell me if this is just a waste of time. It's important clinically, but you guys might already know all this. So here we have a threshold stimulus and we have an action potential. And if we would stimulate the membrane. So here we have this first action potential. Even if we stimulated the membrane with a superthreshold stimulus, we can't generate a second action potential right there. The membrane won't allow it. It turns out the H gate is closed, and until it opens, we can't generate another one. But let's say we wait just a little bit longer. We stimulate the membrane right here. If we get past this point right here and we stimulate the membrane, we can get another action potential. But it takes more stimulus to do it. Why? There's a transient increase in threshold. So yeah, the membrane's still relatively resistant to the action potential, but you can generate another follow-up one. Now, if you generate another action potential right here, guess what? You've started another absolute refractory period, and you've reset another relative refractory period. So let's go over here. What if you wait even longer after that first action potential? Yep, you can generate a second action potential, and it won't take as much stimulus strength because the threshold is now starting to go down back to its normal resting threshold level. And that goes on and on. But once again, if you have an action potential, you suddenly have another brand new absolute refractory period and new relative refractory period for this period following that one. If you wait just a little bit longer, yep, another action potential, but it doesn't take much more stimulus than originally to get the second follow-up one. But once again, anytime you have an action potential, you have another absolute refractory period. How you doing with that? Okay. And so let's say you have an absolute refractory period here. Well, guess what? If you have the same type of situation, yeah, you can do another follow-up. But you reset to an absolute refractory period and you reset to that elevated threshold. Now your book does show that if you, and this is correct, and this is something that you can do at the lab bench, physiologically and clinically, does it have some relevance? Yeah, but your book does show that early on, the amplitude of the action potential is lower. It's lower here than it would be if you stimulated the membrane out here. And that is true. But I really want you to understand the concept of all or none. And when you show the membrane's giving you everything it's got. And I've just kind of exaggerated things here to illustrate the concept. And yeah, the amplitude is a little bit lower here, but it's still an all or none response. It's 100%. This is really what I want you to remember because of some things we're going to be going through later on. Yes?
So do you have to get back to the normal threshold for it to then have that, the full action potential to get back to the absolute refractory period? Do you
Yes.
It has to go back to normal before you have to have that period again?
Which?
The absolute refractory period.
No. You can, so if we have a second follow-up action potential right here, which is occurring in relative refractory period,
Yes.
that is going to generate a brand new absolute refractory period.
So then
Okay?
after the.
So each action potential, it independently will cause its own absolute refractory period. Yeah.
So then does that mean that there's cells that will wait, I guess, for the refractory period to be completely done to get a full action potential?
Are you talking absolute or relative?
Both.
Okay, let me just preface this by saying different cells have different lengths of absolute and different lengths of relative, okay? And so that means different tissues So in relative, you can generate a second
Right.
action potential, right? Different tissues, some may take less stimulation, some may take more.
Okay.
Now, getting to your question again,
Yeah. If, are there cells that wait for the entire refractory period, the absolute and relative, to get a full amplitude?
yes,
Okay.
yes. You, in reality, you do not get the absolute full amplitude until you get very late in the
Right.
relative refractory period.
Right.
That is really kind of a lab bench phenomenon. I mean, there's cases where it can occur in the body, but it's really much more of a lab bench phenomena. And there's a lot of things you can do on the lab bench that your body kind of goes, we've got it. We're going to try to avoid that situation. You know, so. Okay, so let's talk about an action potential from start to finish. So here it's just resting, everything's happy. The membrane depolarizes, you know, maybe we're at the axon hill. So what's going to happen here is the membrane depolarizes up to threshold, and that's going to cause a first voltage-gated sodium channel to sense, oh, we've hit threshold, it's going to spring open. So now sodium's going to start rushing through the open voltage-gated sodium channels. And think about it. Here the first one opens, there's voltage-gated sodium channels next to it. And when that first one springs open, it depolarizes the membrane because sodium is influxing. That depolarization is now going to cause the adjacent voltage-gated sodium channels to now hit threshold themselves and spring open. So from here to here, there is an explosive opening of voltage-gated sodium channels. When one springs open, that causes the adjacent ones to spring open, and then so on and so forth. It's a chain reaction type situation. It's occurring right here. So that membrane is depolarizing, it's racing toward the equilibrium potential for sodium, which is plus 61. That's its direction. It might not get there, but someplace, and I can't tell you where because it's different in different cells. Some cells have voltage-gated sodium channels that when they sense, oh, we're at -10, they begin to close. Other ones begin to close at 0, other ones begin to close at +10. Here we have a case where there's some overshoot. We went past 0, okay, which is not uncommon. So, what we're going to see in the slides to come is sodium channels spring open very rapidly and explosively, but they close kind of much more slowly. They're much more slow to close, and that can lead to this overshoot. So at some point, those same voltage-gated sodium channels that sprang open at threshold, they sense this depolarization and they say, we've done our job, we're going to begin to close. And they begin closing. Simultaneously, there's another set of voltage-gated voltage-gated channels, the voltage-gated potassium channels. They sense, hey, this membrane is depolarized. And that is their signal to open. Now, they don't explode open, they just kind of swing open. So at this point, the sodium channels are closing and the potassium channels are opening. When those sodium channels are closed and the potassium channels are open, then we get this repolarization phase. And you might, in some cells, get short hyperpolarization. Other cells, you won't get it at all. So nobody can really ask you any specific about that. And now we're returning back to our normal resting membrane potential. So, do look at this and just think about this a little bit. When the first voltage-gated sodium channels spring open, that causes sodium to come in. Sodium's carrying that positive charge. So when you have an increase in membrane permeability for sodium, that is going to cause sodium to come in, and that's called conductance. Anytime you're moving a charge, it's conductance. So you can say that you have an increase in membrane permeability, that's an increase in sodium conductance or sodium charge. What's that doing? It's depolarizing the adjacent voltage-gated sodium channels to threshold. So they spring open. So now, basically, this one causing this to spring open has caused this explosion in nanoseconds, a chain reaction of voltage-gated sodium channels to open. So this is considered a positive feedback mechanism. And it causes all these channels to open very, very quickly. So the way it looks something like this, and you do have an illustration in your book on this, and you kind of have to look at this one step at a time. So let's say right here, we've stimulated the membrane. If we go just to this point to threshold, the membrane has just achieved threshold. That's when the voltage-gated sodium channels open. So what this graph right here is the relative permeability of the membrane to sodium. And it's given a value of 1. The membrane is, everybody else is not permeable to sodium. But here at threshold, suddenly the membrane very rapidly becomes very, very, very permeable to sodium. So there's this rapid increase in sodium permeability. But at just about this point, those sodium channels recognize we've done our job. The membrane is depolarized, begin to close. So very quickly, not quite as quickly as the depolarization, but pretty quickly they begin to close, and the permeability for sodium goes down quite rapidly. Not as rapidly as what it was when it went up. At the same time, what do we see? Those potassium channels have just recognized, oh, the membrane's depolarized. Let's start opening for potassium to efflux. So there's this rapid sodium influx, and then those sodium channels close. And just when those sodium channels are closing, those potassium channels begin to open. So this is looking at the relative permeability of the membrane for sodium and potassium. And remember from the slide earlier, for most cells in the body, the resting membrane for potassium is 50 to 100 times greater for potassium than sodium. So if you give resting sodium permeability as 1, the relative resting permeability for potassium is 50 times higher because of those loop channels. Now you can see that there is a 500-fold increase very rapidly in membrane permeability to sodium. That's why you get a rapid depolarization. When you look at the increase in membrane permeability for potassium, it's only about 6-fold. It goes from 50 to 300. But as we go farther along, sodium permeability rapidly goes down, potassium permeability starts to go up. So there's this sequence, one following another, one following another. Increase in sodium permeability, then a decrease in sodium permeability, and an increase in potassium permeability. And then that increase in potassium permeability is going to allow that repolarization. And yep, there's this is the slide I was mentioning you saw earlier, that resting membranes are 50 to 100 times more permeable for sodium. So take a straight edge and kind of go through this kind of thing and go, okay, there's a sequence in permeability changes that bring about a whole action potential. Now, let's see if I can do this in 6 minutes ago. I'm 3 slides from where I wanted to be. How do these sodium channels actually work? Sodium channels actually have 2 gates. There's an M, or activation gate, and an H, or inactivation gate. Your book shows this differently. Your book shows this differently. What do my illustration and the book's illustration have in common? They're both wrong. We're trying to draw something simple to explain something that is occurring in 3 dimensions. But this is very simple and will help you understand things. So here we have a resting membrane. And you can see the M, or activation gate, it's closed at rest. But the H, or the inactivation gate, it's wide open. But you only need to have one gate closed and the channel's closed. Does that make sense? Okay, so let's say something causes that membrane to depolarize to threshold. That M, or activation gate, springs open. Then sodium can go from an area of high concentration to low concentration, causing the membrane potential to rush toward the equilibrium potential for sodium, which is plus 61, so that membrane begins to depolarize because of sodium flowing in the cell. And once again, we're talking millivolts, it's actually very few sodium molecules flowing. Doesn't take many. So, sodium's going to flow, and at some point, this channel protein recognizes the voltage, says, hey, this membrane is depolarized. It's time to close the H, or inactivation gate. That M gate springs open, that H gate swings shut. It's not as rapid. And that's why you can get some overshoot. Now, when does that thing actually close in different cells? In some cells it might be -10, some it might be 0, some it might be +10. It depends on cells. In this example, it's closing at 0. And then the membrane's going to depolarize, repolarize due to the potassium efflux. And once you get down to the resting membrane potential, you get the restoration that, and this does happen in a very specific sequence. First, and I think I have it, first the M gate closes while that H gate is still closed, and then the H gate springs open. Now that is important, because you, as long as that H gate is closed, it's not until that H gate, let me explain it this way. Absolute refractory period begins with the opening of the M gate, and the absolute refractory period does not end until that H gate springs open. Once that H gate springs open, you're in the relative refractory period. And I think I hope that I gave you this line, and you don't, instead I give you that bottom line. Okay. And hopefully I gave you that as well. Now, why do you worry about these things? Well, for example, local anesthetics. Local anesthetics. That very protein right there, local anesthetics block it. They go in, and from this side, it blocks that channel. So it doesn't matter if the M and H gates are wide open, local anesthetics block that channel. Guess what? More depolarization, no action potential, decreased neural traffic, less pain. So potassium is so much simpler because it's only one gate. It's only one gate. Whoa! I'm going to shut the time. I will see you tomorrow for those of you who want to. Which one?